It's an honor for me to introduce Carl Zimmer, a science writer and author who needs no introduction, but here we go. Carl is the science columnist for the New York Times, where he has contributed stories since 2014. Over the past year, he has been one of the paper's leading reporters on the science of the COVID-19 pandemic. The latest of his 14 books is Life's Edge, The Search for What It Means to Be Alive, and Coronaviruses Get Special Billing, which we'll hear more from Carl about. Carl has written for notable publications such as Discover, National Geographic, Wired, The Atlantic, and Stat. And he's won numerous awards, including the National Academy's Communication Award for his previous book, She Has Her Mother's Laugh, The Power, Perversion, and Potential of Heredity. He's also a sought-after speaker at universities, med schools, museums, and festivals, and you've probably heard him on programs like Radiolab and This American Life. Carl is also a professor adjunct in the Department of Molecular Biophysics and Biochemistry at Yale, where he teaches science, he teaches writing. And I'm sure that Carl will say, on top of all those accolades, the highlight of his career is having a species of tapeworm named after him. I will hand over to Carl now, and I'll invite attendees to turn your cameras on if you feel comfortable doing that, and also begin putting questions in the chat for Carl, even while he's giving his presentation, and then later on during the Q&A. All right, so Carl, over to you.
Thanks, Ernie. It's great to be here. I wish I could be with you all in Florida. You know, Connecticut in March has certain drawbacks. So anyway, but here we are, and it's good to be here with you virtually, at least. Of course, the reason that we're speaking virtually is because of this this coronavirus, SARS-CoV-2, which has changed everybody's life on Earth. So I wanted to talk about what this past year has been like for me as a journalist. It's been pretty much exactly a year, I think, since I started working mainly on reporting on the coronavirus. I also was finishing up my book, Life's Edge, at the same time, which was also an unexpected challenge. So it's been an interesting year. And I just want to sort of share with you some of the things that experiences I've had learning about this terrible yet fascinating virus. So I'm going to just share my slides. Give me 1 second. Okay, Zerny, can you give me a thumbs up if that looks good? All right, excellent. Okay. So yeah, so this virus, SARS-CoV-2, no, it didn't even have a name at the beginning of 2020, and now it is, you know, basically the subject of daily headlines, and it has, you know, infected hundreds of millions of people, killed millions of people. It is, you know, there's been really nothing like it in over a century, really, since the great 1918 pandemic. This has, in terms of the speed with which it has taken over the world and turned things upside down, it's pretty astonishing. You know, I am a science writer, and so you know, I am not writing so much about, say, the politics of the pandemic. You know, the debates over how best to sort of buttress society to deal with it, or vaccine distribution. I have really focused on the science of this thing. And, you know, at first, you know, the only science we had was a few cases of pneumonia in China that were being reported at the very end of 2019 and then the beginning of 2020. But then scientists isolated the virus. They figured out it was a coronavirus. So this was related to the coronaviruses that give us mild colds, but also related to dangerous coronaviruses. For example, the coronavirus that caused SARS in 2002, 2003. So once this new virus, SARS-CoV-2, started to really show us just how dangerous it was going to be, taking over China, spreading to other countries, scientists began to really get to know it incredibly well. The speed with which they got to know it was really fast. And so I was writing stories like this one, The Coronavirus Unveiled, which came out in October, to give people a sense of the basic biology that we were learning about this virus. Again, at a speed that really was unprecedented in the history of virology. So what you're seeing here is actually arguably the most accurate representation of the virus. It was created by Ronnie Amaro and her colleagues at University of California. And what they've been doing is sort of building this virus back up on a computer atom by atom. They're using information that comes from experiments in which scientists take parts of the virus, proteins, and crystallize them, and that allows them to get a good look at their structure. They can also use other techniques to use microscopes to be able to see these proteins and parts of the virus in full. And then they go to the computer and basically put these things in there. And the thinking is that you'll be able to actually simulate the coronavirus just using, basic laws of physics. So this virus is about 100 nanometers across. I believe like 10 trillion of them would weigh less than a raindrop. And if we dive in here, you see the surface, the viral membrane, which is a mix of sort of oily molecules and proteins. This is what's called a spike protein. Those blue things are little sugars that help to hide it from the immune system. And at the very tip, there are these three structures that can be opened and closed. So they're called RBDs. And here you're sort of looking down at the spike. They're all closed. They can also flip open. And, like a video like this is obviously beautiful and mind-blowing that science can create something like this. But it's really important also because it helps us with some very practical questions, like how do we get sick with this coronavirus? So here you're seeing that spike protein and that tip there, that light blue part, it's called a receptor binding domain, RBD. And when it flips open like that, it can lock on to a protein on the surface of cells in our airways and in other organs. And so it's this exquisite, beautiful sort of lock and key type system, which is the first step in the virus getting inside of our cell. This is like a simplified version from a piece that I worked on with the graphics editor at the New York Times, Jonathan Coram. So this was actually just, about a year ago, we put this together. We just wanted to give people like the basics, here are some of the key things to know about what this virus does. We sort of found that like in adapting journalism for a pandemic, there's no point in just getting lost in the detail. Let's focus on the things that really matter to help people to understand what these viruses do. So here you see the virus and that protein on the surface of the virus, it's called spike. It's attaching to this receptor that I mentioned called ACE2. And that triggers a whole series of events that are going to cause the virus to fuse to the cell. And that little squiggle inside there, that's the genetic material of the virus. That's the key thing that needs to get inside of the cell. So the virus gets pulled inside, and that the membrane of the virus kind of opens up, and out comes the genetic material, RNA. Our genes are in DNA. These particular viruses use RNA, which is a single-stranded version. So they have about 29 genes in this little piece of RNA. We have 20,000 by contrast. And so what then happens is that our cell starts to make the proteins for the coronavirus, the spike proteins and other proteins as well that are going to become new viruses, but also viral proteins that are going to do things to our cells. For example, block the immune system. This coronavirus has a whole arsenal of weapons that it's going to use against us, and they are encoded in its genes. And so some of these proteins are going to go around the cell and wreak havoc. They're not going to be part of the viruses that go out to infect other cells. Now, this is, again, this is a very simple way to show what's happening inside of our cells when we get coronavirus. So those two blobs there, those are our own proteins that are reading the viral DNA and making the virus's protein from it. These are the things that normally make proteins for us, but the virus hijacks them and gets them to read its own RNA. Now, it helps to let you sort of see conceptually what's happening, but it's actually not a really great representation of what it would look like in a cell. And that's one of these remarkable things that scientists are learning about the coronavirus and what it's doing to make copies of itself as it makes us sick. So if you were to actually look at inside of an infected cell, and if you could make those combinations of viral RNA and our cellular proteins light up, you'd see something really weird, really interesting. It looks like this. So those green things are actually the viral RNA, our proteins for turning RNA into proteins and into viral proteins. And so if you look at this doesn't look like a nice clean little textbook diagram. It looks like a lava lamp. And it actually really is a lava lamp in a very fundamental sense of the word. The thing is that the virus's RNA and our proteins, when they come together, the RNA has a exquisite structure that triggers a change, what's called a phase separation, so that together they form droplets. So our cells are just full of liquid, but these are these are liquid droplets within the liquid, and that's exactly how lava lamps work. Also, you can see this in salad dressing. This is basic physics that the virus has figured out through evolution. And actually, it turns out that this kind of lava lamp physics is really important for our own cells. This is an illustration from a couple years ago in Nature, where they were just talking about this kind of phase separation, long before the coronavirus came on the scene. And so you get our cells are full of these droplets, and special things happen inside of these droplets, special kinds of chemistry that we depend on for our survival. So for example, our cells will curl up big segments of DNA inside of droplets, and they can keep them there and keep them quiet. And because once you form a droplet like this, it actually sort of acts like the surface of it acts like a barrier. So only some things can get inside and some things can't. There's no hard wall there. It's just this remarkable physics that you see in a lava lamp that is creating these special liquid structures. So scientists have just been learning about phase separation, and now we're seeing that the coronavirus has figured out how to exploit it for itself. So if we make drugs to stop the coronavirus from making new proteins or making new RNA, we have to get them inside these droplets. And if they can't get inside, they're not going to do much good. So there are actually scientists now who are working on drugs that target the virus's droplets. So the cell starts making these proteins for the virus, and it also makes copies of the RNA, those genes. They make new virus genes. And this all happens in this very weird little network here called the endoplasmic reticulum. But what's astonishing is that the virus is able to remake our cells to help it to make lots and lots of new viruses. I mean, one cell can make millions of new viruses. And one of the most astonishing things that just got discovered a few months ago is that this picture actually shows something really astonishing. Basically, you take an infected cell and you freeze it and you just cut it open. And you can see these little circles here. These are bubbles inside the cell next to the endoplasmic reticulum that the virus has made. So the virus is re-sculpting our cells. It's creating these chambers, these new virus chambers. And what's really weird is that RNA goes inside of them. So here we can kind of like look at different slices of this frozen cell, and you can kind of get a sense of it. And when scientists reconstruct it, that green stuff is RNA. The purple stuff are these factors for making RNA. The yellow is like this outside of these chambers, and these weird red things, those are viral proteins that act as channels. They plug into these bubbles that the virus has created. So the virus is storing lots and lots of RNA inside of these bubbles, which can then be used to make new viruses. So somehow, we're not really sure how, the RNA gets out of those chambers I was showing you, and it gets packaged with some of these viral proteins into a new virus. So this is all manufactured by our cells. And then the little bubble that a virus forms around a new virus moves to the surface of the cell and then releases the virus. And again, one cell can make thousands or maybe millions of new viruses through this process in a matter of a couple of days. So you can see how just really paying close attention to one virus is revealing all sorts of things we didn't know, not just about coronaviruses, but about our own cells and the kind of physics that is required for our own life. So we're learning a huge amount just by training the world's attention on this one virus. The hope is that we can also develop medicines to treat coronaviruses better. But along the way, we're getting the sort of fundamental basic knowledge too. Another fundamental thing about life is evolution. And this is an insight that Darwin first captured with this tree that he drew in a notebook in 1837. So Darwin was thinking about different species and how they're related to each other. And he imagined species descending from a common ancestor, kind of like in a family tree. Darwin didn't know about viruses. The word virus wasn't used for viruses for another 50 years after he wrote this. But it turns out that that viruses show us evolution in action in a way that just fits Darwin's ideas remarkably well. And I could imagine Darwin might be a virologist if he was born today. So again, like taking my sort of basic biology perspective on this pandemic, some of what I've been writing about is understanding the pandemic as a chapter in evolution. And so, this virus probably did not exist as a human virus until sometime in 2019, maybe mid to late 2019, scientists estimate. It evolved from another virus, from a progenitor virus. And there are lots of viruses on Earth that can evolve into new things. If we look at the virosphere, like the whole world of viruses, there might be trillions of species of viruses. We've only identified, we've only named several thousand. We've identified maybe a few 100,000. So the vast majority we know is out there waiting to be discovered. And that includes coronaviruses too. So when a coronavirus, adapts and causes a new disease, it has to do some key things. So, for example, it has to evolve the ability to latch on to the kinds of proteins on the surface of our cells. And the tighter it can fit, the better it's going to do at infecting us. It also has to adapt to the life inside of our cells. So it needs to be able to form those droplets effectively, for example. It needs to be able to produce lots of new copies of itself. So this doesn't happen overnight. So you can see here, this is an evolutionary tree showing how coronaviruses, one small part of the coronavirus family tree has been evolving over the past few centuries. The blue person there, that's I mentioned SARS. That was the coronavirus disease that broke out in 2002, 2003. You can see that is closely related to a bunch of coronaviruses in bats. And you can see that SARS-CoV-2 is also related to bats coronaviruses, as well as some pangolin coronaviruses that have been isolated from the bodies of pangolins. So SARS-CoV, which caused SARS, and SARS-CoV-2, they have a common ancestor, maybe around, the Middle Ages, we'll say. You can see those bars are sort of show you the kind of confidence intervals. So there's a range of uncertainty that is being shown here. So within the past few hundred years, you have this split that takes you to SARS and COVID-19. That ancestor just lived in bats. That ancestor virus lived in bats, and every now and then it would spill over into other hosts. So the question is, well, what happened, a couple centuries ago on the way to the kind of virus that can cause a global pandemic so quickly now? And from what we know about coronaviruses, chances are that in the past couple hundred years, something like this has happened. So you have a bat that has coronavirus. Doesn't make the bat particularly sick. They just sort of tolerate it. And then you start to have the virus adapting. It might, for example, have adaptations that let it switch from one tissue to another. So it might be able to move to the same protein that's in our lungs in their lungs. You then have the virus jumping from one species of bat to another. You can then have two closely related species of coronaviruses in the same bat at once. And then actually, as their genes are getting copied and packaged into new viruses, they can get shuffled together in a process called recombination. Then every now and then, some other species picks up the virus, maybe through droppings or a predator eating a bat or something like that. And in some cases, it's just a dead end. In other cases, those other hosts can pass it on. And somehow, and we do not know yet exactly how it happened, either we got it directly from bats or maybe from an animal like a pangolin, don't know. Probably happened in China or maybe somewhere in Southeast Asia. And evolution takes this next step, and then we have SARS-CoV-2. But the evolution did not stop as soon as SARS-CoV-2 became a human virus. And viruses mutate. They're sloppy when our cells make new copies of their genes. And so you can actually see how new mutations arise and get passed down to further generations of viruses. So scientists have been tracking the spread of the virus just by looking at these mutations. Most of these mutations are totally harmless. They don't do really much one way or the other. Roughly speaking, one or two of these mutations accumulate in these lineages each month. But you can see how, when the virus shows up in a new region where it hasn't been before, you can then match it to viruses from a particular other region that shares that mutational signature. And then that can give you a clue as to how did the virus get from point A to point B. So here's another tree, and this is a very specific tree. This is a tree that scientists made just by looking at collecting viruses in New York City during the big outbreak in March. The hospitals filled up, the doctors were taking samples, and once they tested positive for coronavirus, then scientists would say, okay, well, let's take some of these and look at their entire genome and look for all the mutations. And then they could compare those mutations to mutations known from other viruses that had been in other samples in other parts of the world. And what they could see is this evolutionary tree, so an update on Darwin's tree. And they could see that after the early viruses in the pandemic were limited to Asia. The earliest samples were limited to China. Later, like in January, you start to have samples in Europe. And then in February, most of the samples showing up in New York have the same mutations as you're finding in Europe. So in other words, people are getting on planes in Europe and they're flying to New York and they're spreading the virus there. Most of them probably didn't even know they were infected. But that's the story of New York. It's a similar story in Boston and Washington, D.C. and many American cities. This had already become a global phenomenon. And to simply say, oh, this is as the president called it, China virus, was just to underestimate what this virus really was. So I wrote about this last April. This became clear even at that point. In your experience as a journalist, you typically do not expect that an article on, viral phylogeography will get a very angry tweet from the President of the United States. But that was a highlight of this past year, I guess you could say. I think that this kind of villainizing of China was part of a mishandling of the pandemic in the United States. There are plenty of important questions to ask about how China mishandled the pandemic early on. No question about that. But each country has a responsibility to manage the pandemic when it arrives on their shores. And I think this was just this kind of thing was a massive distraction. We're paying the price now, in that we have over 500,000 people in the United States dead. We only have 5% of the world population. I believe we're up to about 20% of the deaths. So we have not handled this pandemic well over the past year. But now, that being said, the Trump administration did put at least $10 billion into supporting research on vaccines, and also in securing advanced purchase agreements, both of which allowed vaccines to get accelerated much faster than they would have otherwise. The average time it takes to get a vaccine from initial design to approval could be 10 or 15 years. And so this worldwide, this happened much faster. I mean, the Chinese were making their vaccines. Vaccines were being made in India. They're being made in Europe. Moderna, Johnson, and other companies were doing them here in the United States. It was a worldwide effort, and remarkably, we could tell even in May that things looked like they were going pretty well. It looked like we were going to have vaccines that worked to some extent. The way these vaccines work right now, the first generation, we explained this in a series of explainers, Jonathan Corm and I, is that they basically teach our immune systems to recognize that spike protein I told you about. So the Pfizer and Moderna, what they do is they take some RNA and they put it in a bubble. That bubble fuses to a cell, and then the cell uses that RNA to make a spike protein. It's a little bit like what the virus does, except that this is not a virus. This is just, all this contains is the gene for the spike protein. So your cells produce spike proteins and display them. And then your immune cells can encounter those spike proteins and say, that's strange. And they can do all sorts of things, like they can make antibodies that can latch on precisely to those spike proteins. In November, I showed you a story from May where we were saying like, oh, things are looking promising. 6 months later, we have efficacy data. We have phase 3 clinical trial data showing that the Pfizer-BioNTech vaccine worked really well, like really well. And so did Moderna. And then we've had a bunch of other vaccines come out with efficacy data as well. Just as a scientific achievement, it's astonishing, and it's based on our intimate knowledge of the molecular biology of this virus. Just a few weeks later, however, there was a big new wrinkle in the virus story, and this just has to do with its evolution. Scientists had made spike protein vaccines for a particular kind of SARS-CoV-2 virus, but SARS-CoV-2 kept evolving. And certain variants arose that got scientists very concerned. Right now, there are 3 or 4 of them that they're worried about. But the first one was this called B117. And this is an evolutionary tree showing you how B117 arose in the United Kingdom, probably in maybe like September, something like that. And you can see that this box here shows you a whole list of mutations. And on the spike protein, you can see there are a whole lot of places where the gene changed, and therefore the structure of the protein changed. So we realized that we needed to shift up and start to focus on these variants and try to convey to people like, well, what does it mean that these things are mutating? And one thing that it means with B117 is that it seems like there's something going on with the virus that it is able to transmit more easily from person to person. It's possible maybe that it can grab more tightly to cells, and like I was showing you initially, it's possible that it's able to sustain longer infections inside of people for reasons that are yet to be worked out. It has all these different mutations, and some of them could be playing a role in making B117 more contagious. So, if you walk into a store and, there's coronaviruses floating around, if it's another kind of coronavirus, let's say there's a 10% chance that you're going to come away infected. Well, if it's B117, there's a 15% chance. Now, that still might be a small chance for you, but you put that in a whole country and bad things happen. This thing grows exponentially. So this shows you in the United Kingdom how just in the matter of a couple months, B117 came to be the dominant variant in the country. Basically, it was hard to find anything other than B117 in the United Kingdom. And this happened very rapidly. And then, just as the original SARS-CoV-2 spread from China, B117 spread out of Britain and went into over 90 countries so far, went across the United States. And again, you see the same pattern. So you see this happening in Denmark, you see it happening in Switzerland. And then in February, I reported on how the data was coming together here in the United States, and yep, the same thing was happening here. So this graph here just shows you, basically the little inset there shows you data showing an exponential growth of it. And if you sort of say like, okay, if this keeps going like this, what are we going to see? And you can see here that they projected that by the middle of March, you'd have over 50% B117 in the United States. This is worrisome for a lot of reasons, because what it means is that the control measures that people put in place for other variants that might work against them might not work against B117. It still might be able to grow exponentially. It also turns out to be maybe 50% more lethal. People get sicker with this. They end up in the hospital more and they die more. So, so just as we have these vaccines rolling out, we have to deal with this. And this is the latest data from a testing company called Helix. This is the real data now, and it's showing just we're well on our way to 50%. It's doubling in every 9 or 10 days. So, so we are going to be living in a B117 country very soon. There are a number, the B117 is not the only variant that people are concerned about. There are at least 3 that are known as variants of concern. It looks like B117 is fine when it comes to vaccines, but there are others that might be able to evade vaccines. And so they're of a special concern now to the CDC, which is now, we reported last month that CDC was going to be spending $200 million to track these variants. Now they've gotten about $2 billion in total. So that evolution that Darwin first recognized is now a matter of national importance and is a huge focus of government policy. In order to get us out of this pandemic, we have to track these variants. So I showed you this tree before, like, how did this get here like this? It's kind of weird. This graph here shows you like an ordinary virus, like at the bottom, it sort of ticks along at a very slow rate. But if you look at B117 at that lineage, it just seems to have a whole bunch of extra mutations that seem to come out of nowhere. And research suggests that these new mutations may be actually popping up inside individual people, particularly immunocompromised people who are infected for months. And it just might be a kind of a petri dish for these viruses to evolve a bunch of new mutations. And when they come out, they're sort of loaded for bear. This is just this is a map of the spike protein, and this gives you a sense that there are certain hotspots around the protein where we need to be looking to see when new variants arise. So just to sort of switch gears a bit, at the same time, just for the rest of my talk, I just want to talk quickly about my book, Life's Edge, which I was writing while I was trying to manage with this unexpected news. And my book is about, life and how we define life, how we draw the borders around life. And so, is the coronavirus alive? And so I write about the coronavirus in my book, if we look at a definition of life, we can sort of ask, well, is the coronavirus alive? Based on what I've shown you about what we know about it, there are a lot of different definitions of life. This one is one of the more influential ones, sometimes called the NASA definition. It was come up with in the early 1990s. So, it's got a pedigree, it's about 20 years old. Life is a self-sustained chemical system capable of undergoing Darwinian evolution. So if you accept that as your definition of life, and you don't have to, you can make your own. The answer with viruses, the coronavirus is, well, kind of, sort of. Is it capable of Darwinian evolution? I just showed you it's amazing at evolution. It has adapted to a new host and then gotten better at making use of us. Is it a self-sustained chemical system? Well, no. I just showed you how it has to go into cells and take the cells over in order to replicate. In fact, scientists have been puzzled over viruses and whether they're alive or not for about a century. You can actually make crystals out of viruses and just put them on a shelf like salt, and then take them down a few months later, put them in water, and they become viruses again. So Gerald Joyce, who helped to create the NASA definition of life, once said, according to the working definition, a virus doesn't make the cut. So this thing that has turned our lives upside down and that can evolve, that has such an intimate way of using our biology, is not alive, according to Joyce. Not everyone agrees with him. And the most interesting version, I think, is from Patrick Forterre, a French researcher. And he argues that once a virus gets into a cell, that cell is no longer the cell it was. He calls it a virocell. Because as I showed you, like the virus kind of turns the cell into something else. And we all agree that cells are alive, right? And yet here's a picture from what I showed you earlier. The virus turns that cell into it restructures it. It creates these chambers I told you about. So the yellow and the blue they're two different layers to each of these chambers has. And inside of those, you have this RNA, the virus's RNA, which is going to be then used to make new viruses. You even have these pores these little red pores for getting the RNA maybe in or maybe out, maybe both. And so this is not your ordinary cell anymore. This is a cell that a virus has remade. And so Forterre argues that there are two different kinds of cells in life. There are our ordinary cells. He calls them ribocells. He's naming them after the ribosome, which makes our proteins. Then there are the virocells, and these are the cells that viruses go into and basically transform them. And now they are something else. Their goal is not to make more cells. Ribocells make cells. Virocells do not. He has this lovely passage. He says, whereas the dream of a normal cell is to produce 2 cells, the dream of a virus cell is to produce 100 or more new virus cells. This is lovely, and as a writer, I appreciate his use of language. It has not won everybody over. There are a lot of scientists who still say viruses are not alive, or one researcher put it this way: they lead only a kind of borrowed life. And in the book, I talk about how, well, maybe viruses are helping us to recognize something about life itself that we do not understand. And there's this wonderful passage, which, from the 1930s, when we were just starting to understand viruses, from Norman Peary, a virologist. And he said, when one is asked whether a filter passing virus, that is a virus you detect by filtering fluid from an organism, when one is asked whether a virus is living or dead, the only sensible answer is, I don't know. We know a number of things it will do and a number of things it won't. And if someone will define the word living, I will try to see how the virus fits into the definition. We are still trying to find that definition. A definition may not actually be possible. In fact, life might be something that as I argue in the book, life might be something we shouldn't be trying to define. Life might be something that we need a theory for, like the theory of evolution, the germ theory of disease, the theory of conductivity. How is it that matter can become superconductive? Well, how is it that matter can become alive? And once we have some of those answers, we might have a better idea of what to do with these viruses that are turning our lives upside down. So that's my talk. Thank you so much for your attention today over Zoom. I know we're all zoomed out, so I'm doubly appreciative that you were able to come to hear me talk, and I'm more than happy to take any questions that Zern might have.
All right, thank you so much, Carol. This was really thrilling. It was such a clear presentation. I loved the graphics. So the way that you explained the science was really understandable to most. And then how you tied it back into the book. And I know many here have read your book or have it. I talked with folks who have it on hold in the library. They're in the queue. And some questions did come in about whether you think viruses are alive, or the coronavirus in particular is alive, and you addressed that nicely. Can you talk about, you discuss what you call the hallmarks of life. So maybe there isn't a definition, but there's a unified theory of life, if we can come up with that. What are some of these hallmarks that you think we could use to sort of say, all right, here's life or here's not life?
Well, there certainly have been hallmarks that biologists have brought up over and over again, and I explore them in the book. I sort of towards the beginning of the book, I'm like, okay, well, there are these things that we generally agree are important to life that we could even call hallmarks. And so let me sort of explore them in particular species that are really amazing at those hallmarks. So for metabolism, for example, like metabolism, that sort of self-sustainingness that people think is important for life, that involves getting nutrients, getting energy from the outside, and converting that into fuel, as it were, into that you can then use to carry out chemical reactions. And then you have this web of chemical reactions to build up the building blocks of life, the proteins, the DNA, all the other things that make us up, and then to break them down when you need to get rid of them. So I think pythons are great for that. So a python will go not eating anything for a few weeks or even months, and then can just suddenly swallow up half its weight in something. That's why, boa constrictors as well. That's why you have so much fun in Florida right now with, these invasive snakes, because they just have an amazing metabolism. And what's most amazing is that, how is it that a snake can break down something in a matter of a couple of days that is just swallowed whole? And it turns out that as soon as it starts swallowing its prey, its body starts changing. Its intestines double in weight. Its heart gets bigger. Its liver gets bigger. All these incredible things happen because it's about to get ready to just take on this food and break it down as fast as possible. It takes a huge amount of energy. So to get energy out of the food, you've got to put a huge amount into breaking it down. And actually, the metabolic rate of a python, say, is about the same as a galloping horse, and it'll keep that rate up for days. So, I look in the book at other hallmarks, evolution, for example. I try to run an evolution experiment of my own with some help from some scientists on bacteria. I think that there's a certain intelligence to life, a certain ability to take in signals and make decisions about what to do in response. And I look at slime molds, which are these wonderful, bizarre, gelatinous creatures that you'll find on the forest floor, which have no brain. Each slime mold is just one giant cell, and yet they can essentially do math to figure out what they should do next. So it was a huge amount of fun, to explore that with these hallmarks, looking at hibernating bats in caves in the middle of winter and so on. But, ultimately, like, those are just hallmarks. It's a little like saying, like, well, what's the definition of water? Like, if I say to you, well, the hallmark of water is that it's wet. A hallmark of water is wet, or a or that it's transparent. I mean, okay, that's fine, but there are other things that are wet and transparent. Are they water too? And when water freezes, is that into ice? Is that water as well? So hallmarks only get you so far.
All right, excellent, thanks. And folks can look in the book. I love your example of the Amazon Molly as an example of the reproduction question. So I have a question here from Les Thiel. He's a political science professor here, and he's director of sustainability studies. And he's working on a book on the meaning and fate of nature and human nature in the wake of synthetic biology, artificial intelligence, and other emerging technologies. And he says, we know that viruses evolve, but do they evolve by way of natural selection like all other organisms? So random genetic mutations that are selected for owing to their fitness benefits in a changing environment. If so, how do they manage such quick and plentiful mutations? It would appear to exceed randomness.
Right. So yes, viruses evolve by natural selection. The reason that they can evolve so quickly, and the reason we're dealing with all these new variants, is that for one thing, they have an incredibly fast generation time. So you can you get sick with the coronavirus, and in a matter of a couple of days, you are starting to make millions of new coronaviruses, and they're going into other cells, and they're replicating over and over and over again, even within your body. And then they get into somebody else, and they're doing it there. So humans have maybe roughly a 20 or 30 year replication time. Viruses, we're talking days at most. On top of that, we have big genomes, with about 3.5 billion genetic letters, as it were, and we have many different genes that are very long. So if we were just to be very sloppy about our copying that genetic information, we would all, like, first of all, we'd all die of cancer very quickly. Second of all, children would be born with all sorts of devastating genetic disorders. We have all of these systems in place to repair mistakes. Our cells are proofreading new DNA all the time. So relatively speaking, we have a very low mutation rate. Viruses have a really high mutation rate, thousands to millions of times higher than us, because they're just, especially these RNA viruses, the ways that cells build new viral RNA is kind of sloppy. Coronaviruses actually have a little sort of primitive way of proofreading, but it's still, they mutate very fast. So they have these tiny little genomes. They can't really get much bigger than that without some ways of repairing their their genes. So yes, so viruses go through natural selection. They're the pinnacle of natural selection, really, because evolutionary biologists can study evolution in exquisite detail by studying viruses. They can study them in their labs, just play out experiments with them.
Thank you, Carl. Journalism student Zach Savitsky wants to know, why is it so important that we have a comprehensive definition of life? Or maybe the question is it important that we have a comprehensive definition?
Well, I think it's important just to understand something about the world. I suppose we can go around without an answer, but it seems like something that's crying out for some attention. Like we can, look up in the sky and say like, well, do we really need to know why the planet goes around the sun? Who really cares? But, like that's important. That's interesting. Understanding, how gravity shapes the universe is just interesting of itself. And then actually like it becomes of a huge benefit to us once we understand something fundamental about how the world works. We know so much now in biology. Like biology is so rich with insights down to the molecular detail, some of which I shared today for the coronavirus. I just find it remarkable that that has not been accompanied by a sort of a clear agreement on a definition of life. There are hundreds of definitions of life, and scientists keep coming up with more of them. Um, and uh, and I will get in the middle of fights between scientists saying, "Oh, that person's an idiot. This is the definition of life. No, that person's an idiot. That's totally wrong." Like how did we get here? I just find that fascinating. It's like saying like chemists not being able to agree on what a molecule is.
Yeah. All right, thanks. Microbiology student Julia Paoli asks, she says, it's clear that viral pandemics can't be entirely prevented. What do you think are the most important steps that societies can take to reduce the risk and severity of viral pandemics?
Back off of the bats and other wildlife to begin with. We have turned this into a human-dominated planet. Seeking our own well-being. That's understandable, but that comes the way we've been extracting things from the planet comes with a terrible price, not just to the wildlife itself, which is going through a mass extinction crisis, but to ourselves. Pandemics are emerging diseases are accelerating. And so we have to find ways to say, okay, we're not going to be disrupting ecosystems so much. We're not going to be putting ourselves, on in the line of these viruses that can then spill over. This is something that's been happening for a century. HIV, for example, spilled over as a result of European colonization of Central Africa and the construction of railroads and logging operations and so on. Like that's how this virus went from being perhaps just an occasional infection of some hunter somewhere hunting after monkeys or chimpanzees to a global pandemic that's killed many, many millions of people. We need to have better monitoring to tell when there are viruses that are starting to spill over. We need to have better epidemiology because the Chinese government could have done a lot to stop this while it was in China, and it appears from reporting that that local officials kind of didn't want to look bad, and so they kept things quiet. And by the time that people were really acknowledging what they were dealing with, it was way out of control. And then finally, like we need to be ready, if possible, with antivirals and with vaccines. And making a vaccine for a virus that you've never seen before, that's not easy. But there might be broadly effective vaccines, universal coronavirus vaccines, for example. And there's research going on those right now.
So a kind of related question is from journalism student Ellen Bausbach, who says, who asked, how do you think the role of species like pangolins and bats in COVID evolution and transmission will affect how human conservation efforts and interactions with those animals go forward?
Well, conservation biologists in particular have been taking this opportunity to say, here's another reason to preserve wild ecosystems and to, another issue is that like pangolins, for example, are so in addition to disturbing ecosystems with like logging, for example, there's just huge amounts of hunting that are going on. So pangolins are a perfect case of this. They're just getting wiped out because in some cultures, the scales are considered to be an incredible medicine, which they are not. But airport officials are just seizing huge bags full of pangolin scales. It's just horrific. If we keep doing these sorts of things, we are putting ourselves in contact with these the viruses that the wildlife have. Now, a lot of these again are going to be dead ends. But if you keep increasing the number of exposures that you're having, you're just ramping up the odds that even if it's 1 in a million exposures leads to a pandemic. We're going to be having more of these pandemics. We should be ready for COVID-25 and COVID-30, I'm afraid.
Jill Case, who's the Director of Research Communications here, says it seems like this has been RNA's moment, both on the virus side and the vaccine side, and wonders what this means for future science.
We'll have to see. I mean, we'll have to see if societies learn the lesson that they should about the role of science in preparing us for these pandemics or for fighting them. The only reason that scientists were able to make vaccines so quickly was that they already had a lot of understanding of these coronaviruses that they've been working on for years. And if you talk to these coronavirus experts, they'll tell you that 10, 20, 30 years ago, like nobody cared what they were doing. And they would really struggle to get those grants that they needed to just find out the basics about these things. We're really lucky that there was a fair amount that was understood about these coronaviruses when this pandemic hit. The spike protein, just the spike protein alone is a marvelous, intricate molecular structure. It takes a while to figure out. It's got these tips that are flipping up and down, and once it binds, then part of it gets cut, and there are these harpoons that spring out. Like, you don't figure that out overnight. That takes years of research to figure out. So if people sort of go back to normal and say like, oh well, we don't need to invest in basic research, then we're setting ourselves up for another fall.
So speaking of those harpoons and all those really cool structures that you depict so well in the visualizations, science journalism student Haley Dansby is asking if you can talk more about the importance of those kinds of depictions in covering the virus and the properties and pathways when you're writing for the public. And how the visual aspect of coverage for COVID-19 has been different from other topics you've covered. And then I'll wrap that in with a question from another student, Manny Rea, who asks about the difference with respect to the online medium and how that has changed the way you communicate about this virus compared to, say, 20 years ago.
Yeah, well, I've always been just a huge fan of trying to merge the written word, which I do, with illustrations and visualizations and so on. And that's back from my old days at Discover Magazine, where we just had printed pages. And I would work a lot with illustrators to create illustrations that would convey the gist of something complicated, just with arrows and circles and so on, and just to get it across. And it's been great working with Jonathan Coram on a whole bunch of different projects, because he can bring his amazing visual skills to these challenges in a way that sort of can complement what I'm doing with writing. Or he will create something, a series of images, and then I will write around that. It's a lot of work. I mean, it's especially a lot of work for him, because you need to make these things that look good. We create printed versions for the newspaper edition. Then we create online versions, something that looks good on your computer, but then also like we need them to look good on your phone too. So um, that's sort of an ongoing process that I don't have expertise in, but it's great to be able to collaborate with the people who are working on this. Likewise with podcasts, podcasts weren't really a thing until recently, and I've been on a few times with the Daily podcast, and there's like another way to work through this information and a way to reach people in ways like one episode they said, okay, we're going to give you questions from 5-year-olds, and I'm like, okay. But it was great because it was like, it really forced me to just sort of, I mean, these were legitimate questions these kids had. Probably their parents were wondering them too. And I had to like say, like, how am I going to answer this for a kid? I can't rely on any jargon at all. So it was quite a challenge. The producers there just turned it into something I think was really, really valuable.
Thank you. So Jack Prater, a student journalist, and he writes also for the independent student newspaper here, The Alligator, asks how you handled such a politicized pandemic. We saw that tweet in your presentation when you're just working to inform readers about the science.
Yeah, well, I try to remember that, like, okay, I have a job to do here. I'm not a pundit. But that being said, like misinformation is my enemy. So, unfortunately, we had a president who was sharing lots of misinformation. That was just one example of many. So in that particular case, I actually just went on Twitter and responded with a thread just saying, okay, what you're saying, Mr. President, is not correct. Here is actually what the article says, and went through it like tweet by tweet, and just hoping at least other people might not get the wrong impression. People are constantly emailing me things and saying like, hey, I just saw this on Facebook. Does this sound right to you? And, it's just astonishing how much intentionally misleading information is out there. Not just about the virus, but or masks, but now about vaccines. So, so it is I see that as part of my job as a science writer. And, I think you can't sort of shrink back and be like, oh, it's not my job to engage with that. I think we have to, because I think, we're not getting out of this pandemic if a huge percentage of people have some idea that these vaccines are going to kill them, when in fact they're demonstrated to be incredibly safe and work incredibly well.
Dr. Angela Lindner, who is our Associate Provost for Undergrad Affairs, is asking if you can describe the partnership that you have with STEM practitioners to accomplish the excellent in-depth writing that you do. And she asks how a university can model and nurture this kind of partnership in its curricular and co-curricular offerings to better prepare students to become better science writers and STEM practitioners. She says, I'm a university administrator posing as a chemical environmental engineer and chemist. And Angela, I'll say, tell them to take my class. And Carl, over to you.
I think that I would recommend that scientists spend a little time, first of all, working on their own communication skills. I teach writing workshops for science graduate students, for example. I think that's a valuable thing. Not necessarily that scientists will be writing their own books or something like that, but to at least understand the process and to understand the challenges that there are to write about complicated things for people who are not the specialists on that subject. It's scientists sometimes, I think, don't appreciate that even their other scientists don't know what they're talking about, because everyone is so super specialized. So how do you step back and imagine what it's like not to know what you know? It's a basic cognition problem. Um, I would say that, that may then help them when, a journalist calls, then they can be more aware of that process of the process of how news gets made, and how can they answer questions in a way that's going to be helpful to ending up with articles that are useful and accurate and so on. In terms of I wouldn't say I collaborate. I just, I read studies, I call people up, I try to understand, from my end, that people are very generous with their time. I'd say in the pandemic, there's the extra challenge that a lot of COVID information is coming out really fast. It's coming out before it's being peer reviewed. So you have preprints. Sometimes people are giving me stuff that isn't even online as a preprint yet. I don't even know what to call it, like a preprint. So what do I do? Do I share this on a pretty big platform, or do I just say like, no, this isn't ready for prime time? So I have, set up relationships with a number of scientists who I can reach out to, sometimes late at night, and say like, what do you think? Does this smell right to you? And people will say like, I think this is bogus and here's why. Or they'll say like, this really convinces me. And, I can then sort of move forward from there. Kind of my own peer review, I guess.
Okay. And these are some kudos for you, not a question. We have someone who tuned in from Maine. She's a science writer, Beth Chagrasulis. And I know you've gotten some awards from Science Teacher Association. She says, Carl Zimmer's superpower is the ability to explain complex science topics in as simple a way as possible without spoiling the science. He's a treasure and has helped many of my students and me through my years of teaching. So kudos to you, and thanks for doing that.
That's very kind. I mean, I think that science teachers have maybe the most important job in this country. So whatever I can do to help, I'm happy to do that.
And a couple remaining questions. So Sarah Bresky, who is a microbiology and journalism student, is asking about your process. Like, how do you find stories and how do you narrow down what you want to write about, especially when the topics are so complex?
I talk to a lot of scientists. I try to get a feeling for what the community is recognizing as significant, and often like, certain themes will come up again and again. And then you're like, okay, I think this is the story to write next. Sometimes, just a preprint or a paper just pops on the scene and everyone goes, oh, wow, well, that's obvious. We got to write about that. But it can be kind of overwhelming, especially now when you have, dozens of COVID preprints and papers being posted every single day. You really have to, I'm finding actually like, just that a lot of what I'm doing with my colleagues at the Times is just saying no. People say like, what about this? And I'd be like, no, like that's that trial on a drug, like look how many people are in that trial, like 20 people. Is it a randomized clinical trial? No, it's open label. Like, just, it's not, this will be proven wrong. Don't bother with this. So in a way, I would sort of turn the question upside down. It's not, well, how do you find the stories to write about? It's like, how do you keep from writing about too much science that isn't really worth writing about?
Mm-hmm. And I think this will probably be your last question, and it's another from Zach Savitsky, and I think it's quite interesting. He says, you cover so many topics. Do you feel a responsibility to retain the knowledge that you get from reporting and serve as a live fact-checked encyclopedia, or does your expertise fade once the article, book, talk, whatever is published?
I will admit there is some fading. I think that's the value. If you become a journalist, it's the value to sort of create an archive of your stuff, so you can kind of go in and be like, oh yeah, I already wrote about this. But I would say that with time, you sort of become there are in every science there are tricky concepts There are concepts that are counterintuitive that you are not going to pick up right away. And you start to understand those concepts at a more sort of visceral level, so that you can just sort of jump into the new science with that grounding. You know, and I mean, the scientists can get this in their own fields, you know, through the process of training, getting a PhD and so on, doing research in one area. The challenge for a science writer is that you're writing about much broader areas. So yeah, it's always good to sort of keep some textbooks on the shelf too, and just crack them open and remind yourself of stuff when you forget.
Excellent. Well, Carl, it's been such a treat to have you here. Thank you so much for coming. And on behalf of the Science Journalist in Residence Committee, which is your official host, thanks again. And a big thanks to everybody who joined us. And you mentioned you showed a copy of your book. Have it here. We do have some copies that are available, complimentary for students, staff, faculty. So if you'd like a copy of Carl's book, drop by the journalism department, 2070 Weimer Hall, and grab a copy. And Carl, do people just have to wait till things get back in person to get it signed, or?
I can, you know, I've been sending people book plates, you know, if they just if they contact me through my website, you know, I'm happy to pop something in the mail.
All right, cool. It's carlzimmer.com.
Yep.
All right, perfect. All right, thanks again, Carl, and thanks everybody for being here with us. Take care.